Hello, we are Project Group 17, and our project is a call to advance genomic research in Africa. So before we get started with the presentation, we just want to give you guys a little bit of background as to why individuals may be hesitant to receive genome sequencing. So genetics has been misused as a means to create a scientific basis for race. Among geneticists, it is agreed upon that race has more social roots in social science than true genetic or biological reasoning. See, race is a social construct which groups individuals based upon geographical location and physical characteristics, not in differences in the genomes. And as this first quote kind of alludes to, it shows that there's no single absolute genetic difference between these perceived races or geographical locations. And furthermore, another reason why individuals may be hesitant to receive genome sequencing is because it may postulate and lead to the conclusion that one race is superior to another, which is simply not true. But as this quote shows, Wade juxtaposes an incomplete, inaccurate account of our research on human genetic differences with speculation that recent natural selection has led to worldwide differences in IQ test results, political institutions, and economic development. And finally, another reason why individuals may be hesitant is because in 2019, a genetic project in Africa to bring accessible gene sequencing to over 100,000 Africans fell through when the Wellcome Sanger Institute, a major genome research center in the UK, signed non-commercialization clauses with universities that it collaborated with in Africa. So essentially to prevent them from capitalizing on the genome sequencing of over 4,000 Africans. However, the company then commercialized and tried to sell the gene chips to Thermo Fisher Scientific, which it now claims it did not.
So now let's talk about the underrepresentation of genetic studies in Africa. It is thought that Africa is likely to be the cradle of humankind. Because of this and a multitude of other factors, African populations possess more genetic diversity than any other population in the world. Despite this, Africa remains largely underrepresented in genetic studies. In 2009, an analysis revealed that of the participants in genome-wide association studies, 96% were of European descent. 7 years later, 20% of participants in the Genome-Wide Association studies were of non-European descent. However, this was widely due to studies being done in Asia. The degree to which individuals of African and Latin ancestry, Hispanic people, and indigenous people were being represented barely shifted. Despite Africa having more than 1 billion inhabitants, only about 2% of the genomes mapped globally are African. Our lack of knowledge regarding these populations has exacerbated medical disparities by normalizing a genetic framework that is largely Eurocentric.
Thankfully, a body of geneticists are working to shift the paradigm, evidenced by the push to bring the study of genomics to Africa. Most notable is the Human Hereditary and Health in Africa Consortium, or H3 Africa, a 10-year initiative launched in June of 2010 by the US National Institutes of Health and the UK's Wellcome Trust Foundation. This initiative awarded research grants to African institutions across 30 countries, providing African epidemiologists with the resources necessary to apply genomics to the study of chronic and infectious diseases across the continent. Likewise, the ongoing 3 Million African Genomes Project, spearheaded by the African Society of Human Genetics, will further expand upon these advancements. The project aims to sequence the genomes of approximately 3 million Africans belonging to various ethno-linguistic and regional groups. Data retrieved from this research should fully represent the full scope of Africa's genetic variation, supplementing the efforts put forth by H3 Africa.
There are many benefits associated with diversifying the genome. A lot of researchers hope that it will reduce global health inequities. If the African genome is accurately sequenced, we will have a reference genome that a patient's DNA can be compared to. This is important because currently, treatments and medications are really based on a European genome, which leads to misinformed and in some cases harmful treatments. An example is African Americans who have a variation of the TRPV6 gene that stores calcium. While this may have been beneficial to their ancestors, who didn't have a lot of calcium in their diet, it's harmful when African Americans consume more calcium, especially as recommended by their doctor, because it can lead to breast and prostate cancers. Since most research has focused on the European version of this gene, this connection is usually missed. A unique perspective is taken by evolutionary biologist Constance Hilliard, who studied the TRPV6 gene and says the answer to diversifying the genetic code is not by sequencing more people around the world, but by sequencing African genomes. Assuming that ethnicity indicates genetic makeup, or that everyone is like Europeans, is wrong. A better way is to analyze a person's DNA and have a precise reference genome to compare it to. Reference genomes are the key.
there's also some potential risks to introducing more genome sequencing to Africa. One of these risks is that incorrect assumptions will be made based off of genome sequencing data, such as associating sickle cell disease with African populations due to a racial dynamic, as opposed to keeping in mind that sickle cell is largely, at least currently, theorized to be a beneficial mutation with some negative side effects against malaria infection, per Elguero 2015. And this kind of assumptions exacerbate perceived genetic differences between Africans and Europeans. Also, as basic healthcare is still inaccessible to many Africans, there's a question of whether discoveries from gene sequencing would translate to improvements in health outcome, especially if you don't have access to a hospital already. Some fear investment in gene sequencing could also supplant funding for other more cost-effective medical investments, such as vaccination attempts and trying to ensure hospital access.
In conclusion, initially, genetics were used to create different races and justify racism. However, now it is used to prove that the structure of race is not based on genetics and that race is a sociological concept. The genomes of non-European ethnic groups, e.g. African, Asian, Native American populations, are not well studied and underrepresented due to racism, despite the African population being the most diverse of all. This has led to disparities in healthcare pertaining to these groups. There is now a push to study African genomics and other underrepresented groups, which would enable scientists to be informed about diversity in health in the near future. However, it will be difficult as we must actively rework the racist precedents set by the history of genetics. In order to successfully conduct research, we must be willing to prioritize African scientists and let them lead the research of their own genomics.